Anyway, as you know, we're in the 89th legislative session for Texas right now, but last year in the 88th session, we were able to secure some additional funding for the University of Texas Marine Science Institute. And with that funding, we created the Texas Gulf Coast Research Center. Anyway, and so the overall idea was really to sort of enhance the coastal research that was done by our department and get our faculty to work with state agencies and also work more with faculty on the main campus. And again, just to improve Texas coastal research. So again, the overall goal was to conduct research to support healthy and diverse Texas coastal ecosystems, revealed through science and safeguarded by an informed society. So as you know, our department here is a little different than some of the departments on campus. So we get some money directly from the state of Texas to carry out research, and then we get a portion of the faculty salaries also then to teach primarily graduate classes, although we also teach some undergraduate classes. So again, we're a little bit different than the departments on campus. We're a little bit more research focused. Okay, so anyway, in terms of the Texas Gulf Coast Research Center, these were some of the goals that we had for it. So we wanted this center to support healthy and resilient coastal marine environments for Texas and the protection and stewardship of our marine natural resources. Also to support innovative research approaches and partnerships to enhance our mission and its impact on Texas, our nation, and the world. To seek solutions to reduce the impact of coastal development on marine environment and resources. And again, we wanted this, all these awards and for our own institute to value scientific knowledge that is produced and disseminated without bias or advocacy. So in other words, I mean, we wanted to make sure that what we did was, you know, we weren't cherry picking data for one side or the other of any sort of an environmental issue, but so that our information would be accepted as being accurate. And then finally, we wanted to support excellence in education programs that develop the next generation of marine science leaders and the public awareness of marine environmental issues. So, you know, public education is something that we really do well here, I think. You know, so in terms of thinking about, you know, the College of Natural Sciences and the departments, there are two of them that really have strong outreach emphasis. One is the astronomy department. They have the McDonald Observatory out in West Texas, which is, they have a NPR radio program. Our department as well, and a magazine, Stargate, I think it's called. Anyway, our department, we have a lot of education here. So with the Mission Aransas National Estuarine Research Reserve, we have an education sector. We do a lot of outreach with public schools. We actually do K through Gray, as we used to call it. So we do a lot of things for adult education as well. We have various facilities for it. And we have our own radio program, Science in the Sea, which our first speaker, Lee Fuman, set up, how long ago, Lee? About 20 years it's been going on. So if you listen to NPR, you can hear Science in the Sea. So anyway, so we set up this Texas Gulf Coast Research Center. And again, one of the things we wanted to do was to have some additional resources for our faculty to do some coastal research right here in Texas. And so we wanted to set up some RFPs or requests for proposals to fund research. But we wanted to try to do some things a little bit different than some of the other funding agencies. So one of the things we did was to set up meetings with Texas coastal resource agencies. And again, this is something we knew from our work with the Mission Aransas National Estuarine Research Reserve. So they had an external advisory council that had members from all the different state agencies and NGOs, non-governmental organizations that work on coastal problems here in Texas. And so we invited them here to a meeting right here in the Patton Center. They all got up and talked a little bit about what some of their research priority needs were. And then we had a social basically after that, where we could get together with our faculty and they could talk about some possible ideas of ways they could help out with some of the issues that some of these state agencies or NGOs had. And then the other thing we wanted to do was to increase are the collaborations between our own faculty and faculty on the main campus at UT Austin. So obviously there's big departments up there with really great expertise, whether it was engineering or other departments in the College of Natural Sciences like biology, chemistry, physics, whatever. Anyway, we wanted to get together with them. You can see here's a picture of one of the meetings we did. So in January of 2024, our faculty, we all went up there. They had written up some little information cards about some of their areas of expertise, and then they got together and sort of thought about different possible research topics that the faculty in Austin with their expertise could then add to some of our own faculty's expertise. Anyway, and come up with some innovative research projects. So we had one set of grants we called Impact 2030. So these were to address the near-term challenges with research manager partners. We funded 7 of those. So these are the ones with state agencies or with NGOs. And actually, our first talk, Lee, is an example of one of these where he was working with Texas Parks and Wildlife on a fisheries-related problem. And so these had funding of up to $250,000 for a period of a little bit less than 2 years. And the nice thing about the funding is that since it's state money going right to the University of Texas, there's no overhead on those funds and there was no fringe on the salary. So that's all spendable money. So we also did some Impact 2075 grants. These are the ones that we did with partners, faculty members on the main campus in Austin. Again, so these are to think about longer term projects and get some of these partnerships going and start working on some of the really challenging problems facing our coast. We funded 10 of these. And then we also did some pre-doctoral awards. So with our PhD students, if they had an idea for, maybe another chapter in their dissertation that they, wouldn't have the opportunity to do otherwise, they could write a proposal for that. And we would give them up to $20,000 to help them do an additional impact on in their dissertation. And we also funded some planning grants and then also had matching money. So for example, with some of the state agencies or Texas Sea Grant, you often need matching funds when you're writing those proposals. So rather than using part of their own salaries for it or other mechanisms for using that matching money, we would give them real dollars that they could use to enhance that project. So we funded 25 projects overall. Anyway, these are just some of the general categories: marine and coastal ecosystems, plastic pollution and environmental degradation, habitat change and resilience, innovative solutions and community engagement, wildlife and habitat studies, and data integration and advanced analysis. So anyway, again, so one of the challenges with this is that this money had to be spent within the biennium. And so, again, we had some really great projects started, but they have a relatively short period of time to accomplish their goals. So basically this biennium will end at the end of August. So our faculty have until the end of August to finish these studies and our students and so on. But as a result of that, it was pretty challenging to try to spend all this money in that relatively short period of time. So it turns out the only other way we could get a little more time to spend the money was we could encumber it. If we could encumber it and do a construction project, we could have up to 4 years to spend the money. Now, any of you that know anything about trying to construct things at the University of Texas know it would be absolutely impossible to build a new building within 4 years. So we went to a private company. One thing I'd always wanted to do is we used to have a research vessel called the Longhorn. We had it until 2006. It needed some major repairs. It really wasn't getting used as much as it should have been. And so anyway, we let the vessel go. But something I've always wanted to have is a research vessel here. Again, this morning I talked about the fact that, I think one of the great things about here is location, location, location. We have a marina, and we found out that a research vessel like the one we want to build could fit there. So Texas Parks and Wildlife had a similar vessel, so we invited them in there. They used it for about a year. And so we knew that we could handle a vessel of this size. So anyway, so this is a rendering of the new vessel that we're building. It's 78 feet long. It's a all aluminum hydrofoil catamaran. So that means, so again, catamaran has two separate hulls, but it has a hydrofoil in it. So when it's moving, it lifts it up out of the water. We had a committee that designed this ship. Sharon Herska was on it, as well as Jean Fay-Liu and David Mowring from the Institute for the Jackson School. Anyway, and so we try to design it something for that anybody at the University of Texas, and presumably anybody within the state that would want to use this vessel would have what they need. So again, and we went out in February and got a ride on one that they just built that was just like this one. You know, a few little minor differences they built for Cal Poly and Humboldt, and it was just incredible. So their vessel had a little stronger engine than ours, but it could do 32 knots. Our old Longhorn could do about 8 knots with a strong tailwind. So this goes much faster. Ours will do about 26 knots, they tell us. The other thing that's great about it is in terms of fuel efficiency, it uses about half as much fuel per distance traveled as a standard sort of steel monohull. So anyway, it's going to be a great vessel. Should have it done in about a year to a year and a half from now. And just looking at it from the other perspective, so you can see you have a big A-frame on the back. We have a J-frame on the side. We have a winch with both the conducting cable and a standard cable. We can take divers. There's a platform off the back to accommodate divers getting on and off the boat. We have a compressor on board so they can refill their tanks. We have all sorts of instrumentation. We have berths for 14 people, so 2 on the bridge for captain and another crew member, 12 below on the first deck. We have a kitchen, a mess, a wet lab, and a dry lab, so really going to be a very capable vessel. And we hope all of you that use research vessels will think about using it. We haven't set a day rate for it yet, but we're hoping we can make it very affordable for people to use, and we'll certainly want to use it here for our research and for our classes as well. So anyway, so that's another source of funding that we use from the Texas Gulf Coast Research Center that will be hopefully helping our institute in the future. Okay, and that's it for me. How? I don't know, anybody have any questions or on any of this? Oh, one in the back. That is a nonprofit. That's a good question. I don't know if there's any limitations on that, but so we haven't decided how we're going to set up. So we're trying to get this funding continued in the future. That has not been decided yet. So it's too late for this round anyway. So, but
access to nutritious food to sustain active.
Our next speaker is Joyce Velos.
Thanks, Kyle. Hi, everyone. Again, I'm Joyce, and thanks for coming to this talk. I'll be presenting some very interesting preliminary results on blue food consumption and how species identity influence the nutrient profile of Gulf Coast fishery species. But before anything else, oops. Okay, there you go. Okay, but before anything else, on behalf of the research team, I'd like to thank the funding for the Texas Gulf Coast Research Center for funding this project, and of course our various partners and collaborators for extending and generously supporting this project in all ways possible in order for this project to progress. So let's start. So coastal habitats are considered to be one of the world's most productive ecosystems. They may be extensive or patchy in our coastal landscapes, but they do provide us with a lot of various ecosystem goods and services like spawning and nursery for various marine life. They protect our coast from storm surges and erosion. They act as blue carbon or carbon sinks, and they provide us with food and livelihood, which brings me to blue foods. So these are basically seafood like fish, invertebrate, and algae that can be harvested or cultured in the marine ecosystem. They are rich in essential nutrients like protein, vitamins, and omega-3s. And compared to land-based sources, they have less carbon footprint because they need very little to no resources like land or water. They provide live. Okay, thank you. They provide livelihood to 800 million people, 33 million of which are from the US, and it is becoming increasingly recognized as a critical food resource, especially for food insecure demographics. And this is very relevant because in high income countries like the US, food insecurity is alarmingly increasing, especially for under-resourced demographics. Now, many inhabitants in the Texas coast belong, especially in Corpus Christi, belong to low-income and underprivileged demographics. And recent data actually shows that 17.2% of its population is actually experiencing food insecurity, which means they don't have enough access to nutritious food to sustain active and healthy lifestyle. And this number is alarmingly high, higher than the state and the national averages. But at the same time, this area is teeming with a lot of productive habitats across its many shallow bays and estuaries, giving coastal Texans opportunities to harness and utilize the nutrients from these blue foods. But at the same time, the availability of these nutrients can vary across species. And at community level, environmental conditions can influence the nutrient profiles. And there's studies suggest that habitat configuration might be affecting trophic pathway, micronutrient composition, and abundance. And this is expected to intensify as anthropogenic threats and climate change affect these habitats, further altering how and to what extent we can harness the nutrients from marine blue foods for human nutrition. So today we ask these following questions. Does the concentration and the composition of these critical nutrients differ among fish species in the Texas Gulf Coast? And if so, what are the causes for these variations? So we did this by taking some muscle samples and morphometric traits by leveraging our collaborations and partnership with TPWD. We also took advantage of a local fishing tournament wherein participating anglers donated fishes for science. And so far we have 158 individuals for this, focusing mainly on inshore species that many residents, coastal residents, have access to and who have also access to lagoonal habitats like estuaries and also piers and jetties. And these species are also the majority of the catches in the area. So after we took the muscle samples, we then proceeded to various nutrient analysis, including fatty acid analysis to take the nutrient profile of each species. We also did bulk stable isotope analysis to determine the diet and trophic position using the delta nitrogen 15. And lastly, we also did compound-specific isotope analysis on amino acids to understand and determine the carbon source and habit use using the delta carbon 13. And lastly, to determine food consumption patterns, our collaborators in UT Austin used this diet history questionnaire hosted by the NIH, which is a web-based tool to determine food and diet supplement intake. They've designed this questionnaire to incorporate validated instruments on traditional dietary patterns and specifically added items that could assess blue food consumption commonly caught and consumed in the Texas coast. They've also utilized semi-structured interviews, and you might have seen this flyer floating around some time ago. So they've used this to discuss household use and dependence on locally caught fish, how or its economic or cultural relevance to them, and also their perceived accessibility to seafood for consumption. So for the results, interestingly, or again, well, at least expectedly, we found species-specific variation in terms of the nutrient profiles, particularly based on the essential fatty acids. So essential fatty acids, they are considered like that because they are needed in various important biological processes, but the animal cannot biosynthesize it in large quantities enough to catch up to its metabolism, and they have to supplement it in their diet. So in this PCA plot, you can actually see that in PC1, there's no apparent trend. Sorry, it explains 25% of the variation in the data, but if you look at PC2, you can actually see clustering, particularly looking at spotted sea trout here in yellow and black drum here in black, particularly looking at 4 essential fatty acids. And if you look closely, spotted sea trout is associated with DHA and black drum is associated with ARA, likely suggesting that they might be consuming prey rich in those essential fatty acids. And also could be that spotted sea trout is consuming prey rich in high trophic DHA prey, while black drum is consuming lower trophic ARA prey. And I'm not sure if you also noticed, but gaff-top sail catfish over here is closer to spotted sea trout, which somehow suggests that its nutrient profile is relatively closer to spotted sea trout, which is interesting and intriguing, at least for me. And furthermore, we also saw that its concentration of these essential fatty acids are comparatively higher than the popular ones, including red drum, black drum, and sea trout, which according to our DH3Q data, they are the most commonly mentioned species, meaning they may be the preferred species for food over gaff-top sail catfish. So that's very interesting. So now we know that we know how biological changes for species, they affect how they consume and metabolize nutrients. So we looked at several factors that could be affecting that, and we start with growth. So this is a series of figures showing you how the variation in patterns affect each species and how growth in terms of total length affects the concentration of these 4 essential fatty acids. And for time's sake, I would just like to highlight some notable patterns. So for black drum and red drum, at least for these 3 fatty acids, they have strong negative correlation, meaning or suggesting that as the animal grows, this essential fatty acid decline. But on the other hand, interestingly, we saw that there's no apparent trend for gaff-top sail catfish, meaning that its concentration or the abundance of these essential fatty acids does not change as the animal grows. Also, we looked at trophic level based on delta nitrogen 15. And I'd like to point out that there's still variation in the data, especially across species. But I'd like to point out that, again, for spotted sea trout, it has consistently higher nitrogen 15, meaning it is consuming diet that is high on the higher trophic level. And also for black drum, its nitrogen 15 is somewhat on the lower level, lower ranges compared to the other species, which actually demonstrates and supports our earlier observation that they might be consuming prey on those separate trophic levels. And again, pointing out that for gaff-top sail catfish, there seems to be no apparent trend and potentially suggesting that it has a very stable, potentially stable diet. And lastly, we present very interesting preliminary data on the CSIA dataset. This PCA plot shows you that the red drum actually has a very unique carbon source set of carbon sources than bathtub sail catfish, meaning they could be utilizing very separate or different habitat in the estuary. But if you can recall the previous nutrient profile a while ago, you can see that spotted sea trout and black drum have very distinct nutrient profiles, but in this figure, they kind of overlap, which suggests that they may be utilizing the same carbon sources, but assimilating it differently. And I'd like to point this out as well, that for gaff topsail catfish, there is this carbon source that is not being shared with the other species. So this might be also something that could be influencing the nutrient profile that we see over here. So, and to summarize everything, we've seen that there's species-specific variability in the nutrient profiles, especially based on the essential fatty acids. And we've seen that less desirable species like gaff topsail catfish have comparatively higher nutritional benefits than popular fishes for food. And these variations could be tied well to growth and diet, suggesting that there may be a role of ontogeny and prey preference for this one. And also we found that there's limited indication that the nutrient profiles are actually determined by carbon sources. But since this is still preliminary data, we hope that as we go along, add data and analyze this, we might see more resolution and more detailed information. As we hope to provide novel insights, because this is, as far as we know, will be the first integrated socio-ecological assessment of the importance of marine blue foods for under-resourced households in the US. And lastly, before I end this presentation, I'd like to point this out that, so this is a catfish, right? Specifically a hardhead catfish. In Texas, at least, they don't get a good rep. Like, people just throw them away. People don't like them, so they rarely get eaten. But imagine if species like this that people don't like and often don't target actually have nutritional benefits in them. And we actually saw that in our data a while ago, that they have very comparatively higher concentration of the essential fatty acids compared to popular ones. And this is very relevant because the healthy eating index scores are positively correlated to this essential fatty acids and negatively correlated with food insecurity, which is expected. And this is critical because according to our DH3Q data, the average respondent does not meet the recommended daily intake of these essential fatty acids. So I'd like to highlight that, borrowing from a friend's words, that maybe what people might consider trash or junk fish may be good actually for eating. And also, if you think about it, maybe it would also reduce discards and help the environment. So thank you.
Our next speaker is Sharon Herska, who's going to be talking about her presentation titled Fine-Scale Transport of Planktonic and Early Juvenile Blue Crab Settlement to Estuarine Nursery Habitat in Aransas Bay.
All right, we missed the recipe, Joyce. You should have given us a recipe for catfish tacos or something so we can try them. All right, I'm going to tell you about some of the work that we've been doing in my lab over the last year with funding from the Texas Gulf Coast Research Center. And this is an image from the Aransas Pass. So we're sitting right here. Lydia Ann Channel is up this way, and this way is the Corpus Christi Ship Channel. And when this funding came in from the Texas Gulf Coast Research Center, I reached out to Mark Fisher and Zach Olson at Parks and Wildlife to ask them what sort of priorities they had for research. My background is in larval fish ecology and stable isotope ecology, but at the time they told me that they were very interested in having studies on the early life history of blue crab, and I had never worked with invertebrates before. But I said, well, you know, kind of like larval transport, a fish, blue crab, let's give it a shot. So as I started getting into it, you know, there's very strong similarities with some of the species that spawn along passes and the coast, and then you have the fish larvae that are recruiting to estuaries in the region, like redfish and croaker, Atlantic croaker, they have the same type of life history. So I'm particularly interested in this part of the blue crab life history. The spawning takes place either near passes or along the coast, and then there's a variety of stages, zoeal stages, before they become a megalopy, at which point they are transported into, if they're lucky, they're transported into estuaries and they settle onto nursery habitat, which are seagrasses or marshes. And this transition can be a bottleneck to population size if there's not enough either supply, so there's not enough larvae being produced, or there's something that limits the transport and the settlement or the availability of nurseries. So this is the stage that I'm working on. And it's a complicated stage because there's a variety of physical and biological processes that are driving the recruitment or the ingress of the megalopy into estuary nursery habitat. Now, the megalopy are pretty small. The carapace length here is about 1.1 millimeters, just to give you an idea. Well, I'm sorry, 1.6 millimeters. And then this is the last stage before they transform or they molt into what would look like a little crab. And the smallest ones are around 2 to 3 millimeters carapace width. So they're relatively small. And we have the pier laboratory here that reaches into the ship channel. It's right out the window here. And it's a really good sampling platform for collecting the megalopy as they're coming into the system. So here's the Gulf of Mexico, and I'm sampling here when we're working in the channel. And then we decided to work in Lydian Channel, which is this area over here in Aransas Bay, back here. And the objectives were to quantify the daily ingress during the fall season, which is the peak of the ingress of these species in the area. And we do that with two different approaches. We have, sorry, this pointer here. We're using this net. It's a very long net because we set it to filter at the beginning of the flood tide, leave it out for the entire flood tide. So we have a cone on it, so it only has a small opening, but it has a very high filtration area. So we can quantify the number of megalopy over each flood tide. And we basically designed a sampling program where we were doing 3 days of sampling during the peak of a flood tide, so with diurnal tides where you have the maximum incursion of water into the system, and then followed that with sampling in the seagrass habitat where we were looking for the smallest crabs. We were also trying out these settlement collectors that have been used quite a bit in crab research, particularly here in the region in Ed Buskey's lab, and we basically designed one just like the ones that he used, and we set 6 of them running along the pier so that we would have some degree of replication. Now, when we were putting them out, we were testing them out initially, we saw there were a lot of fish around the pier, so I'm like, they're probably eating the megalopy. So we also added a small like experiment of cages and without cages. We basically put this trap in a minnow cage to try to minimize the predation. So then we were sampling in the seagrass bed, and we chose 5 seagrass habitat sites. And I wanted to try to use stable isotope ratios as a marker or as an indicator of when an individual had recently arrived to seagrass nurseries from the coast, which is something that I had worked on before with redfish recruitment. And lastly, one of the things we're working towards is examining this relationship between the supply of megalopy and the recruitment of the smallest crabs into the seagrass nurseries. Now, we're working here in Lydian Channel. We have a couple, I'll show you where the stations are. But the logic of using or locating different stations along Lydian Channel was this study, which did a particle tracking model to look at the transport of redfish larvae into the estuarine system. And this is the maximum tidal excursion that they calculated during diurnal tides. So if larvae are coming in through the pass and part of that water is going up Lydian Channel, this is the furthest they would reach based on this model. So here are our stations. We have 2 right in Lydian Channel, a 3rd one which is right around the corner if you're going over here by boat, then one that is in this little nook south of Mud Island, and then right off the tip of Mud Island, where there's also an extension of seagrass beds. We were only sampling in Haliduli rhytii, so as to not introduce the confounding or potentially confounding effect of Thalassia versus Haliduli, so it's only Haliduli. And then we were using these enclosures, which we basically sample with dip nets, and then we use a very large pump to pump out the rest of the crabs in the water, because they do hold on to everything for dear life. So to get a good quantitative estimate, you really do need to use a very strong pump. So we did this over the fall of last year. We did it 5 times, and I'm going to present data from once each month. So these are the plankton net sampling data. As I said, we had 3 days on each of the dates. This is everything we collected during the season, and you can see this is megalopy per flood tide. There were a couple of events, one in September, one in November. We were getting thousands in our plankton net per flood tide. And then we also looked at, as we were processing the plankton samples, it turns out there's quite a few juveniles, very early, small 2-millimeter juveniles, first instar juveniles in the plankton net. So they are actually molting, or a part of the population is molting in the water column before they are settling onto the seagrass beds. And in some cases, this was as high as 30 or 40% of the individuals that we were catching. This is the data for the settlement collectors. So we have the total number of megalopy in the 6 settlement collectors. And when we look at these values, you can see that we got a lot in September, and then we didn't get very many for the rest of the year. These are all the dates that we deployed settlement collectors. So you can see there was a lot of effort here, but we were really not getting a lot of them. And we correlated the total number of megalopy in the plankton net during the flood tide versus those in the settlement collectors. There's absolutely no correlation. So we're trying to figure out what that means. It could be that there's differences in developmental stage, in the sense that in the plankton net, the megalopy might not be competent to settle, so they might not be fully ready to make that settlement step and then metamorphose into juveniles. It could be that the current speed, which is extremely strong in the channel during certain parts of the tidal cycle, is too fast for them to actually latch on to the settlement collectors, which implies that there's some sort of gear selectivity, and it could also be patchiness, like horizontal patchiness along channel. We don't know which one it is. We have to work on that. Now, if we look at the 6 settlement collectors, so C1 was the one towards the end of the pier, and C6 is closer to the jetty. When we look at the average abundance over all the samples that we collected, we see no significant differences. And we did not see a difference statistically whether or not we had cages. So either there's small predators eating them in all of the collectors, or they're just not interested. Larger predators are not interested. We also did this analysis by considering only the data for days when we had a relatively high abundance, and we didn't get significant differences either. These are all the data for the settlement collectors, and all I want you to see here is these arrows over here. So when we do get a lot of megalopy, we were getting them on pretty much across all the pier. So they were, they're in the water column, basically. So it doesn't seem like it's a very fine-scale spatial thing where if you're closer to the jetty, there's more, or closer to the end of the pier, there's more. And then when there's few, there's few. So it samples well, but it seems to be sampling something different than what we catch with a plankton net. Okay, so these are the abundance of early juveniles in Lydian Channel. Over here, the first 2 columns, Lydian Channel, then this point here around into Aransas Bay. This is Mud Island, and this is across the season. So the first column is September, October, November, and December. And very quickly, you can see that the 2 sites that are in Lydian Channel, this one and this one, are the ones with the highest abundances. And these are very high abundances. This is 60 individuals per meter squared. And to be honest, it did take us hours to go through these samples. We were out in the field and doing this to try to get all the crabs out of these enclosures. So we have higher abundances consistently closer to the pass, which is basically consistent with tidal current transport of the megalopy being delivered basically by the flood tides into the seagrass habitat. These are the length frequency distributions. So we have the stations here. The first two are the ones in Lydian Channel. And then we have, this is the last one in Mud Island. And these green boxes are basically the size range of anything below 10 millimeters. So these are relatively small juveniles. And you can look at the abundances. They are much higher. The smaller ones are found in higher densities in Lydian Channel. So again, the smaller ones seem to be settling in Lydian Channel. There is the presence of some very small ones further up, but the majority of them seem to be settling probably in the first seagrass habitat that they find as the current is bringing them in. In October, we see something similar. We have relatively higher densities of relatively small individuals in Lydian Channel compared to the other 3 locations. In November, again, we have this pulse of very small individuals, and we don't see as many. We do see quite a few in Mud Island, and if you'll remember the channel, the ship channel, the dredge channel comes along this way, and then it goes up this way. So these 2 stations are actually pretty far from the channel where the bulk of the water is being transported. And this is October. Again, the higher densities are found in Lydian Channel. So that seagrass, which is not very extensive, it's a narrow patch of seagrass that is on either side of Lydian Channel, lying along the banks, seems to be very important for these very early crabs. Now, in terms of the stable isotope data, I only have some of the data to present today, but the concept here is that as the megalopi, or maybe the very, very smallest juveniles we're catching in the plankton, they're feeding on the plankton, they have a particular isotopic composition. And when they recruit to seagrass habitat, they will slowly and over time start reflecting whatever food sources they are feeding on in the seagrass habitat. And we can basically simulate this using laboratory experiments to look at turnover rates. So basically that's telling us how quickly will this process be happening. And we can do that as a function of time or as a function of size, right? So we're looking, this is the way we expect the relationship to look. This is the relationship that you find for all sorts of organisms, but crabs molt. So they lose a lot of their biomass every time that they molt. And I was expecting something like a step function, you know, that they would somehow make this jump rather than a continuous curve. So we've been running those experiments, and this is one of those experiments. This is the carbon data. We didn't have a difference between the diet that we were feeding them versus the planktonic isotopic composition. So this is really not useful, and we have other experiments we ran in consequence, but let's look at the nitrogen data. So this is when we sample them. We get the megalopi from the ship channel, and then we put them in a rack of small incubators where we control the temperature and salinity and all that. And then we started feeding them Artemia, freshly hatched Artemia. These are still megalopi, the blue ones. And after 3 days, they started molting, or all of them were molted by day 4 or so. And this is the pattern. So they actually do follow that isotope turnover rate pattern that is typical of organisms that don't molt, which is, it's quite interesting. And if you look at, this is in terms of time, and this is in terms of size. So they're making these jumps as they are growing. And it turns out that when crabs molt, they can, the size of the next stage can be very variable. And I think that's probably what is contributing to this shape. But what this is telling us is that the isotope composition is changing relatively quickly, and within a few days it's going to reach an equilibrium with whatever it is that they're eating in the seagrass habitat. But during this period of time, we should be able to infer that they have arrived recently. And if we have growth rates, and we're working on that in lab experiments, that we can estimate when they arrive to the nursery habitats. Now, these are data for September, isotope data for September. And what we did was we took all of the very small ones and we analyzed them whole for their isotopic composition, and we subsampled basically because we had too many. But here you can see that the ones that are less than 10 millimeters, or the very small ones, have a different isotopic composition to this cloud of quite variable carbon isotope ratios for the larger crabs. And the nitrogen data, however, looks a lot cleaner. Here we can see that we have the smallest crabs, they have a signature that is presumably more planktonic, and then they eventually reach an equilibrium to the ones that are larger and also collected at the same location. So this is another station in Lydian Channel, and we can see a similar pattern. This is the station over here. We had fewer individuals, and we don't see such a clear, there's not that many small crabs that have a different isotopic composition. So these crabs might be actually moving up the estuary rather than coming directly in from the plankton. And these are the data for the Mud Island site, where we really don't see, different values when they're very, very small. They seem to be more consistent with the rest of them. So this might mean that what we're thinking is happening is they're coming in through the pass, they're settling onto this area, and then they're moving up the estuary once they have settled and metamorphosed. So we think that so far it's very clear that those seagrass habitats, at least in Lydian Channel, are very important. That's where they're landing, most of them. And we can track them with stable isotope ratios. We're going to keep working on that. We're analyzing some of the stable isotope data for the other dates and other turnover experiments that we have been conducting. And ultimately, we're trying to get to a point where we are going to be able to determine whether there's a bottleneck to population size that is associated with the early life stages of blue crab. And I encourage you to look at the poster that we put together. I've been working on an imaging system to do very fine-scale transport process studies with blue crab, and I'll have a poster of our shadowgraph imaging system up this afternoon.
Thank you, Sharon.